Welcome to the Endomembrane Factory, an advanced nanoscale assembly line inside your cells. Today, we explore the step-by-step mechanics of how macromolecules are synthesized, packaged, and shipped. This is cellular logistics at its finest, showing how complex molecular blueprints turn into functional reality. Inside a cell, organelles are not floating freely in a chaotic soup. Instead, they are partitioned by selective lipid membranes. This crucial boundary allows each compartment, or lumen, to maintain its own unique chemical environment. By isolating newly built molecules from degrading enzymes in the cytoplasm, the cell ensures safe protein folding and tagging. The manufacturing line consists of fixed complexes and mobile shuttles. The nucleus acts as the blueprint archive holding your DNA. The rough ER serves as the ribosome-studded factory floor for primary synthesis, while the smooth ER handles lipids and detoxification. The Golgi apparatus acts as the shipping terminal, and transport vesicles serve as mobile shuttles navigating between them. Production begins when an mRNA blueprint arrives from the nucleus. A ribosome engine reads this incoming code and links amino acids together. Through co-translational translocation, the ribosome pumps the growing protein directly across the rough ER membrane into the lumen. This ensures the delicate chain avoids any destructive contact with the surrounding cytosol. Once inside the lumen, specialized chaperone enzymes manipulate the raw protein chain into its precise 3D geometry. Quality control is exceptionally strict. Misfolded or broken products are aggressively rejected and degraded before they can exit. Approved cargo is then encapsulated as the membrane pinches off into a mobile transport vesicle. The mobile vesicle arrives at the shipping dock and fuses with the cis-Golgi membrane. Inside the Golgi stack, enzymes attach precise chemical tags, like carbohydrate chains, which act as molecular zip codes. Repackaged inside specialized outward-bound vesicles, the sorted proteins are dispatched to the cell surface, lysosomes, or external secretion. The smooth ER is an interconnected tubular network that actively pumps calcium ions out of the cytoplasm. By trapping these ions inside, it creates a massive electrochemical gradient, acting like water held back by a dam. When an external signal opens these molecular gates, a torrent of ions is instantly released to trigger rapid actions like muscle contraction. Lysosomes operate as cellular destructive engines. Driven by ATP, mechanical pumps force protons into the core, dropping the internal pH. This extreme acidic shift activates powerful, previously dormant digestive enzymes called acid hydrolases. Fused waste particles are quickly obliterated by this chemical fire and recycled back into raw building blocks. Think of cellular manufacturing as a strict biological balance sheet. The input is a single copy of an mRNA blueprint. The intermediate processing requires ribosome synthesis, an ER folding pass, and Golgi sugar tagging. The final output yield is a fully functional, target-tagged secreted protein ready for deployment. Cellular toxins act like physical wrenches thrown directly into organelle gears. Ribosome blockers like ricin, anisomycin, and puromycin bind to ribosomes, freezing translation and causing cell death. Transport blockers like Brifeldin A sabotage vesicle networks, forcing the Golgi apparatus to dissolve entirely back into the endoplasmic reticulum. When physical or chemical agents rupture the structural integrity of the lipid bilayer, regulatory fail-safes fail. Without a membrane boundary, the controlled internal acid fire of digestive enzymes leaks out. This accidental leakage bypasses normal systems, causing the cell to undergo rapid, uncontrolled pathological self-destruction known as autolysis. Specialized cells radically warp their internal architecture to master singular tasks. For instance, a single activated plasma B cell acts as an antibody factory, mass-producing up to 2,000 targeted proteins every second to fight infections. To meet this insane demand, nature scales the rough ER and Golgi into a high-capacity industrial complex. To wrap things up, let's look at the diagnostic matrix of the endomembrane system. This chart lays out every major node, from the nucleus to the lysosome, alongside their structural features, inputs, outputs, and primary mechanical actions. It also highlights the specific chemical inhibitors, like actinomycin D or chloroquine, that can completely disrupt each organelle's function.